In this section, we're going to talk about the pentose phosphate shunt. You can see from our general schema of metabolism where the pentose phosphate shunt fits into the grand scheme of things. The main thing to remember about the pentose phosphate shunt is that it produces NADPH. This is a really important concept that you need to understand in relation to the pentose phosphate shunt. NADPH is a reducing agent, very similar to NADH, but it has a fundamentally different role in metabolism than NADH does. Rather than using its reduction potential to drive production of energy, NADPH uses its reduction potential to drive redox reactions in biosynthetic pathways. Some of these biosynthetic pathways include glutathione production, fatty acid synthesis and desaturation, cholesterol and sphingolipid production, and the reduction of dihydrofolate. Now, there are two different pathways by which we can produce NADPH in cells. One is the pentose phosphate shunt, and the other is the citrate shuttle, which we'll discuss in the fatty acid metabolism section. The pentose phosphate shunt is called this because it takes glucose 6-phosphate, a 6-carbon sugar, produced by glycolysis, and converts it through a series of steps to fructose 6-phosphate and glyceraldehyde 3-phosphate while generating NADPH in the process. So it's a shunt that goes from one part of glycolysis through an alternative pathway and ends up back in glycolysis. Now, it's called the pentose phosphate shunt because an intermediate in this is the 5-carbon sugar pentose. Now, there are multiple steps in this process, but again, I only want you to understand the rate-limiting and regulated step, which is the first step in the conversion of glucose 6-phosphate to ribose 5-phosphate, which is a pentose. Now, this is a multi-step reaction as illustrated below, and in the process, it generates 2 molecules of NADPH. This reaction is catalyzed by glucose 6-phosphate dehydrogenase, or G6PD. As I said, this is the rate-limiting and the only regulated step in this process. G6PD is regulated by negative feedback inhibition by its product, NADPH. NADPH competes with NADP+ for binding, so that when the NADPH to NADP+ ratio is high, the reaction is inhibited. And this ensures that the cell only produces the needed amount of NADPH and no more. Now, the ribose 5-phosphate, which is produced by the pentose phosphate shunt, has 2 potential fates. Number 1, it can serve as a substrate for the synthesis of nucleotides. It produces the ribose sugar backbone of DNA and RNA, and that's a very important synthetic function. The other thing that it can do is it can be converted through multiple steps back to glycolytic intermediates. This is the shunt. So you take 3 molecules of ribose 5-phosphate, convert it to 2 molecules of fructose 6-phosphate, and 1 molecule of glyceraldehyde 3-phosphate, both of which are intermediates of glycolysis. Now, there are inherited defects of the pentose phosphate shunt, one of which is described in the following clinical vignette. A 3-year-old boy is brought to the emergency room by his parents. They're concerned because he developed yellow eyes and dark urine over the past 2 days. They also report that he seems extremely tired. Upon further questioning, you learn that the family immigrated from Africa just 2 weeks ago. The patient was subsequently diagnosed with malaria 3 days prior to presentation and began primaquine therapy. Physical exam is significant for scleral icterus, conjunctival pallor, and tachycardia. He's anemic and is jaundiced. His CBC shows he has a low hemoglobin and hematocrit, confirming the anemia. The reticulocyte is high. The indirect bilirubin and LDH are elevated, and serum haptoglobin is depressed. The blood smear shows Heinz bodies and bite cells. The bite cells are shown in this picture. They look like red blood cells with a little bite taken out of them. So what disease does this patient have? This patient has glucose 6-phosphate dehydrogenase, or G6PD deficiency. G6PD deficiency is an X-linked recessive disorder, and it's caused by a mutation in the G6PD protein, which makes the G6PD unstable. Normally, G6PD has a half-life of about 62 days. In G6PD deficiency, the half-life is reduced to approximately 13 days. This disease is most commonly found in patients of African or Mediterranean descent. The signs and symptoms of this disease are generally those of any hemolytic anemia, things like fatigue and pallor, jaundice, and tachycardia. The hemolytic anemia and G6PD deficiency is due to the reduced production of NADPH in red blood cells. The main role for NADPH in a red blood cell is recycling of glutathione. Glutathione is a specialized tripeptide made up of a gamma-glutamic acid molecule bonded to cysteine and glycine. The sulfhydryl group of the cysteine is a really strong reducing agent. So it protects the cell against oxidative damage by reducing peroxides in the cell, as shown in the equation below. The oxidized form of glutathione has to be re-reduced so that it can be recycled and reused. The reaction of recycling glutathione is catalyzed by glutathione reductase, and it requires NADPH as indicated below. So why does this disease only occur in red blood cells? Why don't we see other cells in the body that are affected by this disease? Well, one reason is that red blood cells are particularly prone to oxidative damage, and this is because they're professional oxygen-carrying cells. The other reason is that red blood cells rely solely on the pentose phosphate shunt for NADPH production. The other process by which NADPH can be produced is the citrate shuttle requires mitochondria for its function. And since red blood cells don't have mitochondria, they don't have this pathway. The other problem is that red blood cells can't synthesize new protein. So when the unstable G6PD turns over, it can be replaced by new protein synthesis. So in this disease, you get episodic hemolysis, and it's usually caused by infection or exposure to some oxidizing drug, like sulfonamides or nitrofurans, or particularly antimalarial drugs like quinine, primaquine, or quinacrine. It can also be induced by the consumption of flavamines. The increased oxidation causes membrane defects and precipitates hemoglobin, and the Heinz bodies that you see in this disease are aggregates of hemoglobin which has been oxidized. The defective red blood cells are cleared in the spleen, and this is why you get an anemia in this particular disease. So here's our summary of the pentose phosphate shunt. The input to the pentose phosphate shunt is glucose 6-phosphate from glycolysis. The outputs are NADPH, fructose 6-phosphate, and glyceraldehyde 3-phosphate, which are also intermediates of glycolysis. The key enzyme I want you to remember is glucose 6-phosphate dehydrogenase, or G6PD, which is negatively feedback inhibited by NADPH. The associated disease is G6PD deficiency. This concludes the section on the pentose phosphate.